Hi again, I'm Roxanne Newman, Conference Producer for Analytical Cannabis, and I'm here to introduce the next talk in today's event. Quick Scalable Genetic Tests for Hemp and Cannabis. I am really pleased to have Dr. Sebastian Manacapelli and Mark Joubert joining us today as your presenters. Sebastian holds a PhD in Molecular and Cell Biology, a MS in Biochemistry, and a BS in Microbiology. He has worked in the development and implementation of a variety of in vitro and cell-based assays for over a decade, and received the 2014 Publication of the Year by the American Society for Cell Biology. Mark has been practicing law since 2002, primarily focused on patent law. Mark is extremely successful at resolving many intellectual property disputes between patent, trademark, copyright owners, and alleged infringers, either by litigation, mediation, arbitration, or settlement. In addition to being a lawyer, Mark also holds a Bachelor of Science degree in both biology and chemistry, and a Master's in Molecular Biology. A warm welcome to you, Sebastian and Mark. Following the presentation, we will have a Q&A session, and we would welcome any questions that you may have. Please ask questions using the Q&A tab below the video player. If you experience any technical issues during the presentation, please request help using the Q&A system. I will now hand over to Sebastian and Mark.
Hello and welcome. We are Verne Bioanalytics, and we would like to introduce our company to you. We want to thank the organizers of this symposium for an opportunity to introduce ourselves to you and hope that you will find our presentation both informative and useful in your respective endeavors in this changing and dynamic industry. We at Verne Bioanalytics are aware of the needs and challenges relating to our world's growing need to locate and utilize alternative sources and supplements to strained and limited resources globally, specifically in the areas of food, clothing, medicine, and fuel. It is our belief that cannabis, with its many benefits, will provide some degree of value that will provide a supplement to existing and new markets specific to these name fields and possibly others. And with that, I would like to hand over to my friend and colleague, Sebastian Manacapelli.
Well, thank you, Mark, and thank you everyone for listening to our presentation. We are very excited to talk about some of the innovations coming out our research pipeline. Cannabis can be the answer to 21st century needs, since it can produce from plastics to medicinals. But when most crops have benefited from decades of genetic improvements, cannabis has been kept in the sidelines because of its legal status. Seeds for most crops produce very similar plants with highly predictable yields because of their well-known genetics. But cannabis genetics is poorly understood, leading to varieties with highly variable plants that make management difficult. And the occurrence of male plants can derail predicted yields. People have come with solutions to address these needs, like feminized seeds to reduce the number of males and tissue culture to generate clonal plant lines. But these procedures have their own issues. For example, feminized seeds still produce some males, and clonal lines can be genetically weak and unable to cope with infections. Before I go further in the talk, I want to introduce a couple of topics, so we are on the same page. Genetics is the study of traits or characteristics and how they are passed from generation to generation. Some of the genetic characteristics are visible and some are silent. Visible characteristics make up the phenotype of a plant, which is determined by a combination of what it is in the DNA of the plant and the influence of the environment. This is important because your plants might have interesting characteristics that are silent but can become expressed after the right cross. So, why seeds out of the same cross are different? Most of it has to do with how chromosomes, that are basically collections of genes, are inherited at random into the seeds. And this is represented in the diagram on the right. Parents have pairs of chromosomes, here represented in green and red with two different shades, that have many genes, thousands of genes. These genes segregate into gametes, right here, for example, pollen, that then fuse in different combinations to form seeds. The collection of different forms of genes or particular mutations will determine the genetic makeup of the seeds out of a cross. Now imagine having to keep track of all this information for several generations of plant development to develop a new cannabis variety. This is where genetics can help you. If you know the genetic composition of your plants, you can make faster and more informed decisions. Look at the comparative representation of ancient corn, the left, and modern corn in the right. Look at how much it has changed in 7,000 years. Some of these changes were made through traditional crossing and selection over thousands of years, and some were made with modern technology in the last few decades. But the point is, unless you can wait for a long time, you need to know the genetic makeup of your plant. Examples of traits you might want to test your plant for are the presence of THC and CBD genes. In addition, there are other DNA-based tests that can help you improve your yields. For example, male plant identification and pathogen detection. Now, what is the industry standard for genetic testing these days? The standard is a PCR reaction. This is a test that is predominantly performed in laboratories. You send plant material to the company performing the test, they extract the DNA from the samples and run the reactions with a cycler like the one on the left. The PCR machine then cycles temperatures up and down to amplify target DNA. Now, is PCR something I can easily do myself? The answer, or the short answer, is no. A typical PCR workflow starts with a DNA extraction. Then several reaction components are added to it. For example, the enzyme that amplifies the DNA and short stretches of DNA are called primers that direct the amplification of the target sequence. Further, you will need to get not only a cycler, but also liquid handling equipment. Now, this leads to what are the cons of PCR testing. For one, you have to send the plant in. A lot of growers don't want to have other people identifying the characteristics of a particular variety they have been developing. You also have to wait for a couple of extra days because of shipping to whatever it takes the company to go through all your samples. Imagine waiting for a week or a couple of weeks for a set of results when you need to know if your plants are suffering from a pathogen outbreak. And then there is a cost of it. Company needs to cover the cost of facilities and trained personnel. You could do it yourself, but you have to get laboratory equipment and skilled labor. This is where we came in. Our solution is to give you the option to do your own testing without laboratory equipment or skilled labor. Our innovation in our U-Test product line is that it doesn't require a PCR cycler or training to do your own testing. DNA is amplified at a constant temperature that can be reached, for example, in a kitchen oven at low settings. We also have developed patented technology that allows for DNA extraction in the same tube without the need to reopen it to add any reaction components after the tube has been closed. This dramatically speeds up the reaction avoids potential cross-contamination between samples and the need for liquid handling equipment. Now, how this all look in real life? The process is very simple. It consists of 3 steps. You make a leaf punch with the tube's cap. You incubate the reaction in an oven for a few minutes, and then you look for color changes. If it is yellow, it's positive. If it remains red, it's negative. I want to stress 2 very important advantages of this technology. One, it is highly scalable. It doesn't matter if you have 2 plants or thousands of plants. You can run as many reactions at the same time as you need, since you're not limited by the capacity of the equipment. And 2, the avoidance of cross-contaminations between samples, because you don't have to reopen the tubes once they are closed with the leaf punch inside. How this compares in terms of time to our competitors, you can see it represented here at the top. Our U-Test product line takes minutes to completion, while the options from our competitors take days to weeks, and visual inspections, for example, in the case of male detection, can take months because you have to wait for those plants to develop. So what is the Verne onboarding process? Our first production target is a male identification test that will be ready to ship in a few months. And within a year, we will add chemotype and pathogen tests. We're going to gain your trust and want you as a customer. So in the meantime, if you're interested to know how our technology works on your hands, we can send you a test reaction for you to try on cannabis versus any other plant. And once you're in our system, we'll keep you updated on new product developments. Stay tuned. Back to you, Mark. Thank you all again.
Thank you, Sebastian. I'd like to talk about our team. Our team, the 2 of the co-founders, Dr. Johnson and Dr. Manicapelli, have over 20 years of graduate molecular research behind them. They've written many papers. We also have almost 20 years of legal experience. We have assembled a team of other people to help with the lab. We have a lab manager. We have a CFO who has a CPA. We have a marketing and sales director. We are very equipped to scale up, and we are in position to take the market on. The history of this particular company, Verne Bioanalytics, is less than 6 months old. Although the founders have been in this industry and similar industries for many years, and I would say a couple of decades, we just met for the first time like 5 months ago, and shortly later, we formed our first home lab. Later in June, we founded the company, Verne Bioanalytics, LLC. And just several months ago, we began development of the U-Test, which has been finalized. We now have a formal lab, we have a website launch, and we are looking for people interested, such as yourselves. Questions. Verne is looking for partners. We have a contact site at info@vernebioanalytics.com. We have a summary, a series of tests that are in addition to the U-Test. We have several patents pending already. And to learn more about Verne Bioanalytics, please go to our website at vernebioanalytics.com. Oh, we have questions. And please, if the moderators have questions or we have something from the virtual audience, we'd like to field those now.
Thank you very much, Sebastian and Mark. A great talk. We have already received a number of questions, so we'll move straight on to the Q&A now so that we can cover as many as possible. So the first question we've had in is a nice easy one. What is the cost?
I'd like to handle that. And again, this is Mark Jubert. And right now, the U-test, we are price pointing it, but it's well under $10 per test. We have other tests in development, and as we develop those and factor in our various costs, we'll put that on our website. And question number 2.
Thank you, Mark. And the next question, do I need any special equipment?
You can take that. So you actually need nothing beyond what you can find in a standard kitchen. Unlike our competitors or the tests they offer, you don't need any pipettes, PCR, or any other laboratory equipment. Thank you, Roxanne.
Thank you, Sebastian. And the next question, how accurate are these tests?
So I'll take that too. So if what you mean is how reliable the tests are, we have made sure that they work on different cannabis varieties. We will also be sending out positive controls, so you can know if the test works, but the trait or the pathogen you are looking for is not present. Very much like you have in a pregnancy test where there is a control band to show that the test worked.
Thank you. And we have time for just one more question. Does Verne provide other services?
I'd like to field that one, please. Yes, Verne provides other services. One of them is custom assay development. Another is data analysis. The third is bioinformatic services as well, because we have one of our co-founders has an advanced degree, a PhD in this. And lastly, we have consultant opportunities to our customers or people just interested. You can reach out to us at our website, or as I said previously, you can go to info@vernebioanalytics.com and post your question and we will get back to you.
That's great. Thank you very much, Sebastian and Mark, for answering those questions. Just remind everybody, any questions that we didn't have time to get to will be answered offline as soon as possible. That just leaves me to say thanks again, Sebastian and Mark. We really appreciate your time today, and we'll be back soon with the next talk.